Good afternoon, students. So, in our previous class, we have started which chapter? Cell the unit of life. We have started our bio journey with cell the unit of life. So, from this chapter, we have fragmented this like how we can learn it clearly, right? So, in that, we are done with cell discovery, cell theory, exceptions of cell theory. Cell discoveries are pretty easy that you remember so far, right? Who discovered a dead cell? Robert Hooke. From which plant? An oak tree known as Quercus huber, and he is the person who has identified cell wall also for the first time from that cork cell itself. And who discovered living cell? Anton van Leeuwenhoek. And then who discovered nucleus? Robert Brown. Who proposed cell theory? Schleiden and Schwann. We have also learned on the hierarchy of cellular hierarchy of living organization and hierarchy of ecological organization. Tell me the proper definition of a tissue here, because that statement based we may expect. Tissue, group of cells, common origin, common function. Structural similarity may or may not be present. Okay, so those are what? Tissues. Cell theory is applicable to those type of cells which has a cell membrane, cytoplasm and nucleus. Genetic material, it can be in the nucleoid form or nucleus form. A small question for you as we have discussed cell theory. Is cell theory applicable to a prokaryotic cell? Cell theory, is it applicable to a prokaryotic cell? Yes, cell theory is applicable to a prokaryotic cell because it has a cytoplasm, it has nucleoid genetic material and it has a cell membrane. Is it applicable to all types of eukaryotic cell theory? All types of eukaryotic cell? No. Those eukaryotic cells which doesn't have nucleus, which have xenocytic condition, to those cells cell theory is not applicable. There comes the concept of what? Exceptions of cell theory that we have learnt. And we have learned some basic terms also, right? About protoplast. What is a protoplast? Cytoplasm, nucleus and cell membrane. Combinedly you will call it as what? Protoplast, which is a synonym of a cell. What is a protoplasm? Cytoplasm and nucleus, that is protoplasm, right? So this is what we have learnt, and then we have first started with what? Prokaryotic cell. Just give me 2 minutes. Yeah, these are the exceptions of cell theory. First exception is what, ma? Virus. Virus is first of all not a living organism itself, it is a living entity, infectious agent, and these are all obligate parasites. All viruses are obligate parasites, asked in the recent exam NEET 2026, right? We have seen useful bacteria but never a useful virus. All viruses are harmful, right? And then virus means it just has a genetic material and a protein code. Few viruses will also have envelope. Examples: HIV, herpes virus, all these are the examples. And then enucleated cells include matured RBC, sieve tube cells and tracheates. And then we have come across multinucleated or xenocytic cells like Rhizopus bread mold, commonly known as bread mold, which belongs to which class of fungi? Phycomycetes of fungi. So these are the exceptions of cell theory that you have to remember. And then before going to different shapes and sizes of cells, we have started with prokaryotic cell. What we will do today is, we will learn completely on a prokaryotic cell and we will practice some objective at the end of the session on prokaryotic cell. So be here students. First we have started learning prokaryotic cell with the outermost cell envelope. Cell envelope whose layers are how many? 3. Cell envelope is the outermost envelope of a prokaryotic cell, which is a shield of prokaryotic cell. It has 3 layers. What are they? Glycocalyx, cell wall, cell membrane. All the 3 have individual functions. All the 3 have individual functions, but together they are giving the protective unit of a prokaryotic cell. Together they are being what? Protective unit of a prokaryotic cell. Glycocalyx is different in its composition in different cells. In different types of bacteria, it is different in its composition. Like if you see, it can be in the form of capsule or slime layer. Both of them are composed of polysaccharides and murein. But then if it is capsule, they are tightly arranged into a thick wall. They are present in those type of bacteria which cause infections to the host cell. Capsule type of glycocalyx will be present in most of the pathogenic bacteria. Accepted? They are present in most of the pathogenic bacteria, because capsule ultimate purpose is to escape from the host immune system. Bacteria getting escaped from what? Host immune system because of that thick glycocalyx, which is capsule. If that is loosely arranged, you will call it as what? Slime layer. Slime layer where glycocalyx is loosely arranged. Fine. This type of glycocalyx is present in those set of bacteria which are exposed to the extreme environments, like it can be halophils, it can be thermoacidophils, in any of the areas. So, this will prevent the loss of water and mineral nutrients. Okay. Slime layer will prevent what? Loss of water and mineral nutrients. Clear with this too, na beta? Clear, right? Yeah. And then second layer is what? Cell wall. Cell wall is composed of what? Peptidoglycan, which has proteins and sugar units. Sugar units are present in the form of NAG units. What is NAG? N-acetyl glucosamine units. So, it has peptidoglycan. And here comes the concept of gram positive and gram negative bacteria. Gram positive and gram negative bacteria. This is mainly dependent on the composition of cell envelope. That line is given in the NCERT. Based on the composition of cell envelope, we have two types of bacteria, gram positive and gram negative. But in crystal clearly, if we go interiorly, it is specially dependent on the cell wall composition. In NCERT, the statement is given, it is dependent on cell envelope, whether to decide gram positive or gram negative. But in depth, it is dependent on what? Cell wall composition. That is, more the concentration of peptidoglycan, those set of bacteria are what? Gram positive. Less the concentration of peptidoglycan, those set of bacteria are what? Gram negative. Got it right, ma? And third layer is what? Cell membrane. Okay, we learn on gram staining. As of now, you remember this. And third layer is what, ma? Cell membrane. Cell membrane has some extensions which are known as invaginations. Okay, those are known as what? Invaginations. These invaginations can be in the form of tubules, it can be in the form of vesicles or lamellae. Tubules, vesicles or lamellae. Okay, and they are addressed as what? Mesosomes. What are mesosomes? Extensions or invaginations of cell membrane towards the cytoplasm in the form of tubules, vesicles or lamellae. Got it right, ma? These mesosomes have so much of diversity in the functions. See, these are the mesosomal sacs. Can you observe? This will not be asked in your exam, but it is important for your understanding. If we look into the microscopic view, and this red layer is what? Cell membrane or plasma membrane, from which the mesosomes have come. Tubular extensions these are. And you can see mesosomal sacs here, right? From mesosomal sacs, some particles are being secreting. These are what? Respiratory enzymes, which will help in the performing respiration function in a prokaryotic cell. Yeah. Now we will look at the functions of mesosomes. Take down parallelism if you are writing anywhere. If you are having some sticky notes, pasting it in your NCERT also fine. Wherever you feel like an added point, you can take down. Functions of mesosomes. First and foremost function is it will secrete, that means secretion. Secretion of what? Respiratory enzymes, wherein it is also performing the activity of respiration. aerobic and anaerobic type of respiration. Okay. As it is aiding or helping in the process of respiration, there is a most noted term many times asked in your NEET, NCERT also, that mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Apart from secretion and respiration, it also has many other functions. Can you name any of them? Very good. Cell wall formation, DNA replication. Very good. Cell wall formation, DNA replication or duplication, one and the same. And then formation of daughter cells. Most of the prokaryotic cells or almost all of the prokaryotic cell are unicellular. Accepted, agreed with that statement. Most or almost all the prokaryotic cell are unicellular. Okay. So, there is one more statement I am giving. All the unicellular cell or all single celled organisms will divide via binary fission. Accepted, binary fission means if this is a single cell with cytoplasm and somewhere genetic material in the form of nucleoid, this will form a furrow like this. Just without the help of any spindle apparatus or spindle fibers, it will just form a furrow. Cytokinesis will happen, karyokinesis will happen. This is the simple form of unicellular division that happens in all the single celled organism, and the name of it is what? Binary fission. This is how two new cells are formed. This is binary fission. Also known as amitosis, because it is not involving any spindle apparatus for karyokinesis or cytokinesis. Understood, right? So, this is how you will see cell wall formation is happening, DNA replication occurs, formation of daughter cells also happens. This is just by formation of what? Cell furrow. The coordinated or simultaneous activity of karyokinesis and cytokinesis both are happening without spindle apparatus, and this is with the help of what? Mesosomes. How? For example, if you consider this as a cell, prokaryotic cell, and these are mesosomes. Mesosomes from two opposite ends, they will come close together forming out what? Cell furrows like this. Okay, wherein that forms a cell wall, wherein DNA material is duplicated, ultimately daughter cells are formed. And from the mesosomal sacs, they will secrete respiratory enzymes. There how it performs the activity of respiration. Clear? Understood all the functions of mesosomes? Questions on the mesosomes are n number of times asked in your exam. If we can take 10 years of your questions, and if you got 3 to 4 questions from prokaryotic cell, in that 60 to 70% the questions will be from mesosomes only. That important this concept is. Okay. And there is one more point here. Have you taken this, everybody? Yeah, that mesosomes in blue-green algae. Blue-green algae is a prokaryotic cell or eukaryotic cell? Huh? Very good. Very good, ma. I will take some time to register your name. Yeah, blue-green algae is also known as cyanobacteria. It is prokaryotic. Very good. How did you decide that it is prokaryotic? Simple. The basic difference is a prokaryotic cell genetic material is not surrounded by a nuclear envelope, whereas a eukaryotic cell nucleus is surrounded by nuclear envelope, right. That is a basic difference where we will classify any organism whether it is prokaryotic or eukaryotic. Very good on your answer. So, blue green algae or cyanobacteria, if you see, they also have the cell membrane extensions. If I have to take nostoc, which has some heterocyst also, nostoc if I have to take, and this is the cell membrane, this forms some mucilaginous coating over it. I am going to draw only cell membrane extensions. If these are the cell membrane extensions in the form of tubules, these extensions are known as chromatophores. Okay. There is a line or statement in your NCERT that mesosomes in cyanobacteria are known as what? Chromatophores, or cell membrane extensions in cyanobacteria are known as what? Chromatophores. These are known as chromatophores. Chroma means what? Colored, because it has colored pigments. Colored pigments like bacteriochlorophyll and carotenoids. You can take down this. Bacteriochlorophyll and carotenoids. These pigments present in the chromatophores will help in the photosynthetic function. They will help in light dependent reactions. Okay? They will perform what? Light dependent reactions in photosynthesis. Okay. This light dependent reactions that happen in the chromatophores will help in the conversion of physical energy into chemical energy. That is, it will convert photons into chemical energy, that is ATP and NADPH. Done. This is the story of chromatophores. Have explained you all this because we will come across this in plant physiology again. Okay? Very good. Students from virtual platform also. Someone asked what is lysosome. Lysosomes are those cells or particles, secretory particles, which are released from trans phase of Golgi. Yeah. Someone answered prokaryotic, good. Fine. Done with chromatophores. So, you are clear on mesosomes, which are the membranous extensions in bacteria. You are clear on chromatophores, which are cell membrane extensions in cyanobacteria, and some photosynthetic bacteria as well. Cyanobacteria and photosynthetic bacteria. Photosynthetic bacteria examples, can anybody tell me? Purple or purple and green photosynthetic bacteria, or sulfur bacteria, green sulfur bacteria, those comes under what? Photosynthetic bacteria. Okay, clear on chromatophores. And then now we are done with the cell envelope: glycocalyx, cell wall, cell membrane, mesosomes, chromatophores. Now entering into the cytoplasm. Cytoplasm is the main arena of all cellular activities. That is a statement given in the NCERT. Clear, right? Because all the cellular activities are performed inside the cytoplasm only. When it comes to prokaryotic cell doesn't have any membrane bound organelles. It has only non-membrane bound organelles. So there is no active compartmentalization of cytoplasm. Yes or no? Whereas in a eukaryotic cell, you can see the compartmentalization of cytoplasm by membrane bound organelles. Here, no compartmentalization. What are those non-membrane bound organelles present, and what are the components of cytoplasm in prokaryotic cell? First is genetic material, beta. First is genetic material. This genetic material is of two types. Of course, in a prokaryotic cell, genetic material is not covered by a nuclear envelope, but then genetic material is not scattered all across the cytoplasm. It is at one place, right? That is bounded by a nucleoid, that is known as what? That region is known as what? Nucleoid. Okay? That is not a complete nuclear envelope, but that is a small layer which is keeping the genetic material at one place in the cytoplasm, right? Apart from that genetic material, there is an extra chromosomal DNA known as what? Plasmid DNA. Got it right, ma? So here genetic material is of two types. The first type is known as genomic DNA, or chromosomal DNA, or nucleoid DNA as well. These are the synonyms. Question can be asked with any of these terms. You should not get confused. Okay. And second is extra chromosomal DNA, extra chromosomal DNA or plasmid DNA. This is about genetic material. Got it? We have two types. One is genomic DNA, nucleoid DNA or chromosomal DNA. The second part is extra chromosomal DNA or plasmid DNA. Okay. Here, genomic DNA or chromosomal DNA will take care of all the metabolic activities of the prokaryotic cell. This will take care of all metabolic activities of prokaryotic cell, whereas plasmid DNA again has diverse functions. Okay, plasmid DNA again has diverse functions. Plasmid DNA is responsible for specific phenotypic character of the bacteria. We as a human beings, looking at us, people will say, yeah, he or she is a human being, because we all have same kind of phenotypic gestures. Same like bacteria also has, but all of us are variable in our phenotypes. Likewise, bacteria is also variable. That variability is from what? Plasmid DNA. Plasmid DNA will decide the phenotypic character of the bacteria. It gives specific phenotypic character to bacteria. Okay, it gives specific phenotypic character to bacteria. Plasmid DNA is responsible for antibiotic resistance as well. Plasmid DNA responsible for antibiotic resistance. Fine, right? And plasmid DNA will have certain factors like resistant factor, fertility factor. Likewise, fertility factor is known as F factor, which is present in the plasmid DNA. That is responsible for transferring of the genetic material, which is conjugation. I think you people already know that what are different types of sexual recombination in a prokaryotic cell. It can be through transformation, it can be through conjugation, it can be through transduction. Of these three processes, we will remember or recall conjugation now. Wherein conjugation, if you see, what is conjugation? The direct transfer of genetic material from one bacteria to other bacteria via conjugation tube or sex pillars. Like how that bacteria is decided, if you see. Let us suppose this is one bacteria and this is the other. One bacteria which has F factor inside it is known as F plus strain. Okay. The bacteria which doesn't have F factor is known as F minus strain. Okay, this is F plus strain and this is F minus strain. F plus strain, this bacteria will help in the transfer of genetic material by extending what? Its pillars or pili, which is a non-motile structure of prokaryotic cell. This is known as conjugation tube or sex pili. After the transfer of genetic material, this will help in the formation of both F plus strains. So, at the end we will get F plus daughter cells. This is the main genomic DNA. Got it? If at all the transfer is from genomic DNA, if on genomic DNA you have F factor and this F factor is being transferred to the plasmid DNA, that is known as HFR. HFR strain means what, ma? Here I will be writing it. Yeah, suppose this is the genomic DNA. Over the genomic DNA you have the F factor and this F factor will be transferred to the plasmid DNA of F minus strain. Okay, from HFR to F minus. And what daughter cells you will be getting here? What are the products of this? The products of this includes HFR and F plus strain. Got it? Once again should I repeat? Okay. Plasmid DNA will help in the performing three functions which are described in our NCERT. First is it will provide the specific phenotypic character to the bacteria. Okay, it provides specific phenotypic character to the bacteria. Second is antibiotic resistance from the bacteria, and third is conjugation. Conjugation have, here you see, the type of bacteria which has F factor on the plasmid DNA, you will address it as what, ma? F plus strain. Those bacteria over the plasmid DNA, if it doesn't have F factor, you will address it as F minus strain. The transfer of F factor from F plus strain to F minus strain, that products what you will get? F plus and F plus. Got it? And if at all the F factor is present in the genomic DNA, not on the plasmid DNA, it is present where? In the genomic DNA. But it will transfer the F factor to the plasmid DNA itself. Okay? This is addressed as HFR and this is F minus strain. And the products you will get here is HFR and F plus strain. Got it? These are little application based questions where you will get, if you look into your previous NEET papers, hardly 1 or 2 questions are from the application part of conjugation like this, from F plus to F minus and HFR to F minus. What are the products you are going to get from here? Okay. So, we are clear on the cell envelope, we are clear on the genetic material. Now, we are going to move into the cytoplasmic non-membrane bound organelles. Yeah. So, you are clear with this, visible to all, right? Yeah. Non-membrane bound organelles. We have two non-membrane bound organelles. Can you name them? First is ribosomes. Second are inclusion bodies. First we will learn on ribosomes. Ribosomes are the reticular structures, small reticular structures or granular structures. These granular structures are discovered first by George Palade. George Palade. First described by whom? George Palade. Okay, ribosomes are first discovered by George Palade. They are of what size, beta? 15 nanometers by 20 nanometers of size. And ribosomes are composed of what? RNA and proteins. Ribosomes are composed of RNA and proteins, and that is the reason ribosomes are known as ribonucleoproteins. They are also known as ribonucleoproteins. Got it? Ribosomes discovered by George Palade. They are of 15 nanometers by 20 nanometers in size, and then they are composed of what? RNA and proteins, known as what? Ribonucleoproteins. Now, if you see at the sites of ribosomes and what are the subunits of ribosomes? Prokaryotic cell will have 70S ribosomes and it has two subunits. Okay, it has 70S ribosomes and it has two subunits. Those two subunits include 50S ribosomes, which is larger subunit of 50S ribosomes, and smaller subunit is of 30S. Okay, ribosome is of 70S. Any doubts, Kanda? You can ask me. Any doubt, ma? What is that point? Composition of ribosomes. It is composed of RNA and proteins, which is known as ribonucleoproteins. Clear, right? Yeah. And here you see ribosomes in a prokaryotic cell are of what? 70S ribosomes, whose larger subunit is 50S, smaller subunit is 30S. What is this S here? Swedberg's unit, known as sedimentation coefficient. Okay, when the cell is centrifuged, when the constituents are centrifuged, you can see this ribosomes at what sedimentation coefficient? 70S. But then larger subunit and smaller subunit are individually found at 50S and 30S subunit. This nobody will ask you, but then you have to know why is this 70S and individually it is 50 and 30. Their association will make them condense and they are at what? 70S subunit, and this S is Swedberg unit. Clear, right? And then where are these ribosomes present in a prokaryotic cell? They are present inside the cytoplasm, and few of the ribosomes are attached to plasma membrane as well. Few of them are attached to plasma membrane as well. Here if you observe, of course the ultimate function of ribosome is what, ma? Protein synthesis. The ultimate function of ribosome is protein synthesis. Ribosomes being attached to cell membrane or they are present freely in the cytoplasm, their function is protein synthesis only. But those ribosomes attached to the plasma membrane will also aid or help in translocation of proteins. They will aid or help in translocation of proteins. Okay. Here we need to get used with the term of polysome or polyribosome, right. What is a polysome? To a single mRNA strand, mRNA which is formed after transcription, to a single mRNA strand, multiple ribosomes are attached. Multiple ribosomes are attached. Over the mRNA, ribosomes will be attached, and with the help of tRNA, proteins are being synthesized. We know that. In a eukaryotic cell, if you see, over a single mRNA, single ribosome is attached. That will give you single protein expression, that is monocistronic protein expression. Whereas in prokaryotes, many ribosomes attached to single mRNA will give you multiple protein expression, that is polycistronic protein expression. That is the key difference between prokaryotes and eukaryotes, asked multiple times. This is a connected question asked along with molecular biology. Okay. So, prokaryotes and eukaryotes. In prokaryotes, you can see single mRNA strand with multiple ribosomes. This condition is known as what, ma? Polysome or polyribosome. Where each association will give one protein expression, which is multiple protein expression. That is known as what? Polycistronic. Whereas in eukaryotes, if you see, over a single mRNA strand from 5 prime to 3 prime, single ribosome is attached, which is addressed as what? Monosome. Okay, monosome. This will give monocistronic expression. Got it? This is about ribosomes. Where are they present? Of what subunits? How are they composed? What will be their size? Okay, where are they actually located on cytoplasm and attaching to plasma membrane? And what is the phenomena of polysome or polyribosome? Clear on ribosomes completely? Clear, right? And next non-membrane bound organelle you have to learn on is about inclusion bodies. If this I have to suppose like a prokaryotic cell, and this is the complete cytoplasm, right. So, here inclusion bodies are the storage bodies without any membrane. Okay, they are the storage granules without surrounded by any membrane. They are membraneless storage granules. Examples of inclusion bodies includes phosphate granules, cyanophycean granules, and sulfur granules or sulfate granules as well. Okay. Some complex form of inclusion bodies will also be present, like poly beta hydroxybutyrate granules. Not required, I am just telling you. These are the simple granules which are membraneless, present in the cytoplasm. Till here you remember. In some of the prokaryotic cell, gas vacuoles are also present, which will aid in buoyancy. Got it? Clear till here? Yeah. So, this is all about a prokaryotic cell. We have learnt on cell envelope, then genetic material, followed by ribosomes, and then inclusion bodies. Last but not the least, we are left over with motile and non-motile structures of prokaryotic cell. Motile and the only motile structure of a prokaryotic cell, can you see the diagram? The only motile structure is what? Flagella. There is no cilia beta. There is no cilia, it is only flagella. Flagella is the only motile structure of the prokaryotic cell. Pili and fimbriae, these are the non-motile structures of a prokaryotic cell. Flagella is composed of a protein known as flagellin. The only motile structure is what? Flagella, composed of flagellin protein. If I have to suppose this as cell membrane and this is the cell wall and this is the glycocalyx. Okay. Flagella will originate from cell membrane and these are the extensions of cell wall. Okay. Flagella originates from cell membrane and these are extensions of cell wall. Fine. And then you have three regions or parts of cell membrane, which includes basal body, hook and filament. Basal body, hook and filament. Basal body is the structure which is adjoined to the cell membrane and cell wall. It is adjoined with some rings like this, which are LSMN rings, not required, the names are not required, but remember basal body adjoined here with rings. And then you have hook and filament. This central portion is known as what? Axoneme. If I do a transverse section of this flagella and observe, you will see the microtubular arrangement of the flagella. How do you think the flagella will move? Like this. Flagella will be moving like this. That is because of microtubular organization. How the microtubular organization is there? The organization is like this, where you will learn when I am telling you about centriole also. At the center there is a proteinaceous hub. Okay. To this 9 radial spokes are present. Each radial spoke has 3 peripheral microtubules. Again they are arranged like this, 3 peripheral microtubules. Okay. So on total it is what type of microtubular arrangement? 9 radial spokes. At the center there is no microtubule, it is what? Proteinaceous hub. So it is 9 plus 0 arrangement of microtubules. 9 plus 0 microtubular arrangement. Got it right? And then you will observe about non-motile structures. Non-motile structures include pili and fimbriae. First is pili. We have learnt about sex pili or true pili, which is present mostly in gram-negative bacteria. Gram-negative bacteria. Okay. How you can observe pili as? They are again the tubular extensions from the cell wall. Okay. They are tubular extensions. Their main function is adhesion. Okay, where they can attach to the substratum. Sex pili or conjugation tube will also aid in conjugation, that is transfer of genetic material. And second is fimbriae. Fimbriae are nothing but bristle like structures. These are what? Tubular structures like this. Okay, whereas fimbriae include bristle like structures which will sprout from the cell membrane. Okay, clear till here about motile and non-motile structures. Last 5-10 minutes we will be doing some objective. No problem, you can take down, I will save it. Okay. Give me 2 minutes. Meanwhile, you can revise the entire prokaryotic cell. Yeah, so here we are. Just give me 2 minutes. Done? Can you see this? Difficult, right? No problem, leave. This you answer. First question. To online students also. Assertion. In prokaryotes, mitochondria are absent, correct or not? True. In prokaryotes, mesosomes are present which help in respiration, true or wrong? Out of these 4, which you will select? Speak out, not with your friend, with me. Augustine. Assertion is? In prokaryotes, mitochondria is absent, correct. In prokaryotes, mesosomes are present which helps in respiration. Both the statements are correct, but reason is not explaining assertion. Option 2. About this I need to still explain you. Yeah. I hope you can answer this diagram based. From your NCERT, direct diagram from your NCERT. 2? We'll see. Think. 4 types of cells A to D, A, B, C and D, shown with their relative cell size. If these 4 structures are bacteria, eukaryotic cell, PPLO and virus, then which of the following is correct? Firstly, this is eukaryotic cell, this will be bacteria, this is PPLO, one type of mycoplasma, and these are viruses. Now you tell me, B is bacteria, A is eukaryotic cell and C is PPLO. Option 2 is right. Next. You can answer this. Consider the following statements. Inclusion bodies in prokaryotic cell are bounded by single membrane. Correct or not? Okay. Second, gas vacuoles are inclusion bodies in purple and green photosynthetic bacteria, which will aid in buoyancy. So, statement 1 is incorrect, statement 2 is correct. Option? Next. Cell theory is done right, you can answer. What happened? Schwann, what did he say? Why this much thinking? He will feel bad. Select the right option which relates Schwann regarding the following statements. He reported that cells have a thin outer layer which is today known as plasma membrane. Cell wall is unique character to plant cell only. The bodies of plant and animals composed of cells, products of cells. Matthias Schleiden has said only plant body is composed of cells. It is Schwann who said both plant and animal bodies are. Yeah. Both of them said the same point.
No, products of cells.
Yeah.
Given by
Rudolf Virchow has said on how a new cell is formed. How a new cell is formed. The plant and animal body are composed of cells and products of cells are said both by Schleiden and Schwann. Schleiden said it is from a plant body. Schwann addressed it is both in plant and animal bodies. Okay. So, all the statements are correct. Both of them didn't say how new cells are formed. Then, second question. Last question actually of today's session. Tomorrow we will practice few more. Online students also you can answer. We will see who will answer first. Very good. It is the extension of plasma membrane in the cytoplasm in prokaryotic cell. It helps in cell wall formation, DNA replication, register all the activities, okay. Helps in respiration, secretion, increases cell surface area of plasma membrane, correct. It is the characteristic of only bacteria cells. What are they? Mesosomes. Got it? Clear? Tomorrow we will solve few more questions on sizes, shapes of the cells, and then we will also move into cell wall and fluid mosaic model, that is cell membrane.